Okay, we're going to be taking a look at phylogeny and systematics, using diagrams to help us see the differences and the relationships between organisms. A lot of these diagrams are going to try to examine the evolutionary relationship between organisms, and you're going to see that it's much more useful than just using generic artificial ways of classifying organisms. You should be familiar with the human genus and species classification of Homo sapiens, but you know that that's actually just a tiny piece in a full classification of kingdom, phylum, class, order, family, genus, and species. There are actually subfamilies, suborders, subclasses, and it gets really, really complex. And somewhere there's a big dictionary or online encyclopedia where you can find all of these things. There's various acronyms you can remember, right? King Philip came over for good spaghetti, and there's all kinds of different ones. You need to know the full classification of humans. For kingdoms, obviously we are animals, so it's Animalia. The phylum is Chordata. The class is Mammalia for mammals. The order is Primates. Family, Hominidae. And the genus is, of course, Homo, and the species is Sapiens. Usually, when we're writing down a genus and species name of any type of organism, we don't really have to write the full classification, just as like the first and last name, Homo sapiens. Usually it's italicized, the genus is always capitalized, and the species is lowercase. Other examples are like Homo erectus, Homo habilis, and the famous bacteria E. coli, Escherichia coli. So E. coli is the genus and species name. All right, that's the full classification of humans. Make sure you know that. Also, in the ecology and evolution unit, in the regular part of the syllabus, you're supposed to know 2 full classifications of other organisms. That is strictly a memorization thing, so just do your best with that. There's plenty of examples. I think I know the blue whale and the sunflower full classifications. Good luck with that. There are various reasons for doing classification, obviously to help us identify species. If you're a bird watcher, you go out, you have a book, and you look at various characteristics that helps you with species identification. Predictive value, if B is in the same group as A, then B will probably have or share some of those same characteristics. Evolutionary links are important. Species in the same group share characteristics because they evolved from a common ancestor. So if we're looking at the pentadactyl limb, for example, we can see that all of them who share this pentadactyl limb form evolved from a common ancestor, and it helps us to draw some conclusions and understandings about that. Artificial classification has very little value besides simple identification. For example, insects, birds, and bats all belong in the same group because they can fly. That's just an example of an analogous structure. It doesn't help us to draw further conclusions and find out, on the chemical level, for example, protein sequences and DNA sequences. It's very superficial, and it doesn't help us to understand our evolutionary origins. When talking about evolutionary relationships, it's important to point out, with all the research that we have now, just how closely related we are to other living organisms. We've seen this over and over again. For example, all living things use DNA and RNA as the genetic material with the same A, T, C, G type of universal code. It's called the universal genetic code, and every 3 of those bases is called a codon, and it codes for an amino acid. We all use the same amino acids, 20 amino acids. You don't have to name all of them, but there are only 20 we use to make all the proteins in all living organisms' bodies. On top of that, if you're studying organic chemistry, then you understand a molecule like this can have a mirror image. They cannot be superimposed on each other. It's just different ways to construct the same molecule. It seems like the same molecule, but its mirror image will actually make its properties different, and people have received Nobel Prizes about understanding this relationship. It turns out that all living things only use the left-handed version of molecules, even though right-handed molecule versions could be present. All living things are using the left-handed versions. We can construct this in the lab, and turns out when we actually make drugs, we have to be very careful about which version of those drugs. We can make them in 2 different styles. We call them the left-hand version and the right-hand version. They are mirror images. But all living things use the left-handed amino acids. How cool is that? Do we have to say any more? So, if you actually look at all of this together, if you can put this into an evolutionary tree and put it in a map and show how different organisms are related, it's very, very enlightening. Phylogeny is the main term we're trying to go through. Tracing evolutionary links and origins is called phylogeny. So the phylogeny of many groups is studied by comparing protein sequences and DNA sequences. Remember that a banana shares 50% of the same genes as human. That's ridiculous. No one would ever have thought anything like that. So the similarity between, for example, the amino acid sequence of the hemoglobin gene, for example, we can find out that humans and chimpanzees, how close their hemoglobin gene sequence is compared to like bovine hemoglobin and pig hemoglobin. And from there, we can actually count up all the differences, the various mutations that have arisen over all these years of change and evolution. And then we can develop like an evolutionary clock and actually put in order and figure out where in history these various evolutionary changes and new species have arisen. So when we put them into a diagram, we're going to see this in the next few slides, but when we put them in a diagram, you can actually map out approximately when humans split off from their most recent ancestor. So clades, cladograms, and cladistics are all discussing the exact same thing. So when you see a tree diagram like this, the nodes, the branching points, show related groups. These related groups are supposed to have a common ancestry. So each group is called a clade. You could just say one of these groups is a clade or this group is a clade, but they have to have the same origin. You can't have two separate pieces and not include the rest of the piece. So a clade is a group of organisms that evolved from a common ancestor, and this information is constructed using molecular differences, basically DNA sequences, molecular differences, protein sequences. And these tree diagrams that are showing these clades are collectively called cladograms. So they're used to reevaluate the traditional classifications of organisms. So we're actually using molecular data. That can be a strength and a weakness. The strength is it's very objective. It's totally based on collected molecular evidence and data that can be repeated by different groups. The weakness is that many of these molecular differences are based on probability calculations when you're sampling different types of organisms that are out there. So the full definition of cladistics, if you go study this in college and decide you want to become a statistician in biology, then one thing you would probably go and study and major in and do a PhD in is cladistics, which is the method of classification of living organisms based on the construction analysis of cladograms. So it's just the same word over and over again, but we're going to see an example of how to actually use this. Here are a few diagrams showing some of this stuff. This is a cladogram of primates, and you can see where the common ancestors are and where they actually started branching off. So humans are right up here, relationship with apes. We separated from tarsiers and lemurs much further back, but this is all based off of molecular evidence. A phylogenetic tree is kind of showing the same thing, but sometimes these phylogenetic trees can also be drawn to scale, or at least to a logarithmic scale, and it gives you a time frame as well too. Really useful. And finally, here's a practical example where you are presented with some data and you're asked to construct a cladogram. After you've looked at this a few times, you're going to realize how easy it is to actually put this stuff together. So if you look at this chart here, you can pause the video and read through everything. But this is showing the presence or absence of various traits. And then we can see here that the mouse has all of these traits: jaws, limbs, hair, and a placenta, whereas a salmon is only showing jaws and is missing some of these things. A jellyfish lacks all of them. So if you take a look at all of these traits and try to put them in order and try to understand that anything that's up here will have to share a trait with everything else that's on this same branch because they're all common ancestors. They're all from a common ancestor. The further back we go, something that exists here does not necessarily have to exhibit the same characteristics as something this far along in evolution. So this is more recent in time, this is going back in time. So if we just look at this, we can actually find out and plot some of these things. The jellyfish has nothing in common with all these other things: salmon, salamander, koala, and mouse. It's just basically looking at the number of plus signs. There's different variations of ways to ask this type of question, but overall, not too difficult to solve. And then understand, though, that this has been provided for you. You didn't have to analyze all of the molecular evidence in order to create this chart, but you won't be expected to.